Welcome, everyone. I'm Kira Curtin, Senior Editor at GenomeWeb, and I'll be your moderator today. The title of today's webinar is Investigating Cellular Targets in Narcolepsy and Other Arousal-Based Disorders with Single-Cell Spatial Transcriptomics and Optogenetics, and is sponsored by VizGen. Our speaker today is Roberto De Luca, a neuroscientist at Harvard Medical School in the Beth Israel Deaconess Medical Center Department of Neurology. You may type in a question at any time during the webinar. You can do this through the Q&A panel, which appears on the right side of the webinar presentation. And if you look to the bottom tray of your window, there are a series of widgets to enhance your webinar experience. And with that, I'll turn it over to Roberto. Roberto?
Thank you very much. It's a pleasure and an honor to be here today, and thank you very much for organizing this webinar. And I'm so excited to present my last result of my most recent research, and it's about the role of orexin neurons that inhibit sleep to promote arousal. So first of all, let's go through the neurophysiology, and for the people that are not familiar with the field, just a quick refresher. So how the neurons communicate to each other. So basically, we have an event called action potential that is like a modification of sodium conductance, so how the neurons get activated and how the neuron can signal what carries in terms of information to another neuron. So the action potential can go through the body and the axon, and it's called a presynaptic input, and will contact basically in electrochemical manner the postsynaptic neuron downstream. And what we study is just this. We study the communication between the neurons and how a signal can propagate and can target the other neuron that is like downstream. So we have a presynaptic and a postsynaptic cell receiving the input. But how this takes place in our brain? So basically we have a wave of propagation, and the action potential reaches the axon terminals and depolarizes the membrane. Then we have the activation of the voltage-gated calcium channels that will open and let the calcium flow going in. And then this calcium influx triggers the synaptic vesicles for the neurotransmitter release. And then from the presynaptic side, we move to the postsynaptic side, and the cell gets depolarized or hyperpolarized, depends on which transmitter we can trigger. And the neurotransmitter binds to the receptor on the target cell, and in this case, cause an activation, but as I said, can cause also an inhibition. So it's like a phone call between two people, can be a positive or a negative, and there is like an exchange on ideas, on emotions. But we are talking about neurons here, so. And what's it, optogenetics? So basically what we saw so far is what happens normally in communicating, so between neurons in the brain, naturally speaking. But optogenetics is one beautiful approach that we can use in the lab to induce an artificial stimulation or inhibition to trigger this communication or inhibit this communication between the neurons. And can be done in vitro, so like studying the inputs between the neurons, or like in living animals to study what optogenetics can do to trigger a change in the behavior of the animals. In this case, we study sleep and wakefulness, so we study the change in sleep and wake behavior. And this was, of course, Karl Dizeroth was the first, together with the others, to combine these genetic and optical methods. And of course, this works with light-sensitive opsins that are proteins that are sensitive to the light. And these proteins will enable millisecond scale precision to keep the pace with fast dynamics and neural events. So basically, when we shine the light, we can get a really fast response where we, induce the expression of these light-sensitive opsins. And of course, this is a deliver of gain function or loss function events. Depends what we are doing, which kind of manipulation we are doing. So to go on and introduce you a little bit what I did in my study, basically we use this approach. So we use these opsins, in this case channel opsin, that is like a channel sensitive to the blue light that can trigger the calcium and sodium influx, so to activate the cell. And we used a packaging of this, construct into a viral construct. So through a viral injection in the brain, we induce the expression of this protein into the neurons. And once we have the expression, we can do our manipulation in vitro or in vivo, and I will tell you more in a few seconds. And mostly we use a Kree-LOX system that it's like a system that relies on a recombination. So we use a transgenic mice that express this recombinase under the promoter of specific genes. And once we have the virus inside, it's Kree-sensitive. It's a Kree-dependent virus that can be, cut, and the construct can be recombined into the recipient neurons. Also, we have other flip recombinase. There are many systems, but in this case, we want to work on a specific transfection and specific expression in a particular cell populations. And of course, we have chosen our opsin. It is, as I said, the channel opsin. We can activate with the blue light and through a virus, and we can induce the expression to the organism in vitro. And this, what I do basically is I do a slice preparation. So I cut the brain in very thick slices, and I record living neuron, and then I shine the light and see how these neurons connect to each other. On the other hand, also in our lab, we developed like an in vivo stimulation. So again, we inject the virus into the brain, and we let the virus expressing the opsin, and then we implant an optical fiber. In this case, there is no slicing of the brain. The animal is living, and we can modulate the activity or change the behavior in case with the light shining into the brain. And the in vitro is based on the study of the functional connectivity. In vivo is like demonstrate of relevance of a specific pathway for a change in the behavior or physiology. So we had like an idea that having this powerful tool in our hands, we can study the connection between the neurons, and we can basically also study the changes in the behavior of the animals. But what about the neuronal expression? So we have to combine different things here. So we have to know the nature of the neurons that we are manipulating, what they can express, what are their connections, what are the inputs, and how the system entirely works. So, and the future of neuroscience is now passing through the spatial transcriptomics. It's a beautiful and impressive approach that can tell us basically many things about the expression of the neurons in the brain. So we can, know more about, if we know like a morphology of a cell, we can also know more about the expression. If we know about the electrophysiology, so the property of the neuron, we can know more about their expression and also their internal connection. And more importantly, if we know a behavior of an animal, we can induce a behavior. We want to know more about what neurons can trigger this behavior. This will give us everything to go through the cell-specific neuropharmacology, because the new area will be the precision medicine. So how we can target a specific receptor and tackle, and that answer to very important questions like about a disease. So if we know the specific receptor in specific neurons, we can design a better drugs to be very specific and without having side effects. So today, my work is like about sleep, and just I want to give to you a quick introduction about sleep and sleep stages. Sleep is like a common behavior found in all animals, also from jellyfish. And we have different stages here. Very quickly, I go through. We have a non-REM sleep that is divided in 4 stages, and we have REM sleep that is characterized by it's a rapid eye movement sleep, and it's characterized by a loss of muscular tone. Non-REM sleep is called deep sleep or like slow wave sleep. And of course, in this panel, you can see here that when we are awake, we have a movement of our muscles. So we are interested in our lab to study basically the changes in awakened sleep, which circuitries are responsible of this switch. And of course, in a disease, like in narcolepsy, what's going on when we have like a specific loss of specific neurons, and how this balance between sleep and wakefulness can be really compromised. So narcolepsy is a disease that is characterized by a sudden and a consistent daytime sleepiness, and is caused by a loss of orexin neurons. And this is a very interesting disease because we can study this disease in the animal models, because mice, rats, even dogs, and humans, they have narcolepsy through the same cause. So the loss of orexin neurons. It's really a perfect disease to study in using like lab models, like animal models. And orexin is like a peptide that is produced by orexin neurons. Is the only source of, this peptide is produced only by orexin neurons. And it's an excitatory peptide because binds to two excitatory receptors. This is very, very important because I will tell you more later why it's important. And the action is like increasing the activity of the neurons through the increase of calcium and reducing like the potassium conductance. So it's like a G-coupled receptor activation. So orexin A and orexin B. Orexin A binds both orexin 1 and orexin 2 receptor, and orexin B binds more orexin 2 receptor. So actually, the action of orexin in the brain is to induce an excitation. And is something that we found that is not always the case, but I will tell you more later. So if we study sleep, we have to study an important region that is called ventrolateral preoptic nucleus. And the ventrolateral preoptic area is contains a cluster of sleep-promoting neurons. And basically, this was found through a projection to another very important area that promotes wakefulness, that is the tuberomammillary nucleus, where the staminergic neurons are. The VLPO is involved in the initiation and maintenance of sleep, and the lesion of this area is proportion to the size, causes a big sleep loss that is proportional to the size of lesion. In our study, we focus our attention on the galanin neurons. The galanin neurons are the black neurons that you can see here in the staining. And these neurons are important in the promotion and maintenance of sleep. So they really represent the function of this area. But I will tell you more later that this area is not only composed of galanin neurons, it's much more complex than we thought at the beginning. And of course, the relationship with this area with other brain areas is impressive. So VLPO sends projection that receives projection from many brain nuclei involved in sleep and wakefulness. And we found an interesting projection from one part of the brain that is called the lateral hypothalamus. And everything started from here. So my project started from how the lateral hypothalamus can modulate the VLPO activity in promoting wakefulness. This is the main question. And how the galanin neurons can interact with other neurons that are sending projection in these areas. So we recently published that there is a group of inhibitory neurons in the lateral hypothalamus that release GABA, and GABA is an inhibitory transmitter, and these neurons send projection to the VLPO. And it's interesting that when we did this experiment in vitro brain slices, I found a strong monosynaptic connectivity that is really directed on the galanin neurons. So to trigger wakefulness, the lateral hypothalamus GABAergic neurons inhibit monosynaptically the galanin neurons in the VLPO. And this is a very strong input because it's direct, is monosynaptic, so it does not go through another neuron, another interneuron. And the activation in vivo of this input rapidly drives wakefulness from non-REM sleep. This is important because now I will tell you about the orexin story that is a little bit different from this story. And basically, in the lateral hypothalamus, we have many neurons. We have these GABA neurons, and we have the orexin neurons, and many more. But it's interesting that to promote wakefulness, they project to the VLPO, but the action in the VLPO is different because we have different neurons. If we are talking about the GABA neurons, to promote wakefulness, the projection should be very strong and monosynaptically directed to shut down the galanin neurons. If we talk about other neurons, we need to consider the VLPO as a more complex organization. So, and then I will move to the region that is like connected also with orexin neurons. So it was found that the orexin neurons innervated the VLPO, and orexin neurons innervate the whole brain, the same projection all over the place. But we found also, it was reported, and also we found in our study that is recently accepted in Nature Communication, that we have a functional input of orexin neurons into the VLPO. As you can see here, this was published studies in which you can see the fibers going through. And also, an interesting study reported that the macroinjection of orexin into the VLPO increased arousal in rodents. So it means that the orexin project to the VLPO, the orexin neurons project there, and the orexin has a strong effect in there to promote arousal, but we don't know the mechanism behind. If you remember, orexin neurons are able only to excite. And if we want to promote wakefulness, we need to shut down sleep. We need to shut down the galanin neurons that promote sleep. So how can be done this if orexin is only capable of exciting? And this was the big finding of our study, because I discovered the mechanism by which orexin neurons can shut down the galanin neurons, but indirectly. To keep in mind, the last piece of information for you, that in the VLPO we have an heterogeneity. We do not have only the galanin neurons that promote sleep. We have also other neurons that are excited by arousal transmitters like noradrenaline. It makes the story very complicated, because we have these neurons inhibited by noradrenaline that are supposed to be the sleep promoting, and we know later that after my experiments that these neurons express galanin. But also we have other neurons that respond differently with an excitation. And also they have in the rat different shapes, different properties. And of course, I can tell you the electrophysiological assessment cannot be sufficient to demonstrate that these neurons are different. So this in the rat, in the mouse is much more complicated. We have common features shared among the neurons. We need to have really a better identification of the expression of the neurons through the spatial transcriptomics, to the single-cell PCR, to the pharmacology. So let's move down to the VLPO complexity. And what we found is that the stimulation of the orexin VLPO input rapidly triggers arousal from sleep. So how we did that? So we used a orexin Cree mouse. This mouse is capable of expressing the Cree recombinase under the promoter of orexin. So we can manipulate only the orexin neurons through an injection of viruses that will induce the expression of these opsins, the channeled opsin in this case. So once we inject this opsin into the orexin field, we checked how this transfection was specific with an immunohistochemistry. We will implant the mouse for the optical stimulation in vivo. So implanting an optical fiber above the VLPO, because it's our target. So we want to stimulate the orexin VLPO input. And we measure the electroencephalographic waves to measure sleep and wakefulness. So what we found is impressive. It's that orexin neurons trigger arousal through their innervation to the VLPO. And we tested different frequencies, and we found that, between 10 and 20 hertz, we have an arousal in mice. So the mice wakes up. And it's not a spontaneous waking up, it's just an inducible waking process through the optical stimulation. And it's important that we can trigger arousal from both stages of sleep, non-REM and REM. If you remember, our previous study was we were only capable of inducing arousal from non-REM sleep, because we were talking about different neurons, different projection, different circuit. Now we have in the same area another neurons type that projects there, but the effect is different. So the arousal takes place from both stages of sleep if we can stimulate in this particular stage. So, and again, what is the mechanism behind? Orexin can only excite, so we want to shut down the galanin neurons. And we found that indeed, surprisingly, that orexin has a dual response in the VLPO. So one type of neuron is excited. You can see the action potentials here, and then the increase of firing frequency during the bath application of orexin. So we apply the drug in the bath and we record from the slice. As you can see here, this is like a brain slice, and we record the neurons and we record the response to the drugs. And also another half of the neurons in panel E, you can see that is really inhibited. So how this can be done? And the answer is in the next experiment, in which I show that basically the inhibition triggered by orexin is indirect and is mediated by the network, so the other connections, and is mediated by GABA and GABA-A signaling. And why? Because if you record a neuron inhibited by orexin, the same neuron, if the antagonist of GABA-A is applied, the inhibition is blocked. It means that the inhibition is indirect. It has to do with the GABA release, and when you stop the GABA-A signaling, the inhibition is blocked. And also, this inhibition goes away in the presence of the TTX, that is a blocker of the sodium channel. So basically, stop the action potentials, stops the electrical network activity, and what you measure is just really your postsynaptic cell. And you can see here in panel E that the neurons excited by orexin, they keep their excitation in the presence of this blocker. It means we have a receptor there. It means that these neurons can get activated in the same manner. But the neurons that got inhibited by orexin, the TTX stops this inhibition. It means that they had to be inhibited by a GABA release coming from an activation of GABAergic neurons. And now we know the mechanism, the inhibition is indirect, and the next question was, is the population of the GABA interneurons inside the VLPO or is somewhere else? And before going to this point, we verified that in a specific labeled group of neurons in the VLPO, we have the same response. So I studied now in this case the GABAergic neurons using another mouse line. And this mouse is like, let me to label the GABAergic neurons specifically with a virus. And I tested the orexin, and I saw again the two responses. So the excitation and inhibition. And this excitation and inhibition on the same neuron was followed by noradrenaline, and as previously published. So noradrenaline is like a pharmacological tool to identify the two groups in the VLPO, the presumably wake-promoting and the presumably sleep-promoting. And orexin is following basically what noradrenaline can tell us. But now we know that among the GABAergic neurons, we have two groups. But what they express, are they different? Are they so different? And the answer is yes. They share similarities, but they are different. So both of them, they are GABAergic, express VGAT, and both of them, they express the GAD1 and GAD2 that are the GABA synthesizing enzymes. But there is one big difference, at least in this low throughput study that I did on the single cell PCR, that one that is inhibited by noradrenaline expresses galanin. So it's the sleep-promoting cell group in the VLPO. The other group that is excited by noradrenaline lacks galanin. It means that this cell here shares many things with this other cell, but the most important thing is that one is galanin expressing and sleep promoting, the other one is galanin negative and presumably wake promoting. So we know now that we have two groups there in the VLPO, two groups of GABAergic neurons. One does one thing, the other one does the opposite stuff. It's like counteracting each other. But we don't know if there is a connection in there. If we don't know if there is like an internal connectivity. And this our next step was to verify basically if these two groups are communicating between each other. And we did this with the optogenetics. So now we start from the pharmacology, the single cell PCR, and we go through the optogenetics. And remember, these neurons that were patched and tested for different drugs, and also I tested singularly for these different genes. Unfortunately, the approach that I used was limited because it's like, 4 different genes, only 4 genes on the recorded neurons. To know more about the complexity, we need to have like single cell technology or like spatial transcriptomics, but we have this later, so I'm gonna go there later. So, of course, I can really move quickly here because I already explained this at the beginning, but this we demonstrated that the GABA neurons in the VLPO that are inhibited by noradrenaline express galanin, and we tested this with the galanin-free mouse. So we tested everything that we proved before with a specific mouse lines. And also we tested the orexin action. We have only one behavior here. We have only one inhibition, so from both orexin and noradrenaline. And also, presynaptically speaking, these neurons are inhibited through a GABA release, of course, because this inhibition by orexin is not direct, it's indirect. So and as we demonstrated before, relies on the GABAergic input. Now, the key experiment that was really the final evidence that we have 2 groups of neurons, 2 groups of GABAergic neurons, and one is galanin, the other one is not galanin. And there is really an internal circuit in the VLPO. The answer is yes. There is an internal circuit because we did with a crossing between 2 different mouse lines, the galanin-free and the gut flip. So we did a very complicated experiment in which we tried to isolate the VLPO circuit here, and we induced the expression of the opsin on the GABAergic neurons and also labeling the galinergic neurons to be recorded. And what I found, I try to be very simple here. I know that this is very complicated, but I try to be very simple, that the connectivity between these green neurons here to this green and red is direct, is monosynaptic, and is very strong. So it means that internally in this region, we have not only the galanin sleep-promoting neurons, but we have also other neurons, GABAergic, that inhibit the galanin. They can be like a switch. They can be a new pharmacological target to treat many arousal-based disorders, including narcolepsy. And we have like an evidence of a very strong internal communication in there. And I tested this with the optogenetics, and I see when I shine the light that there is a strong current probability of evoking current when I stimulate the GABAergic neurons not expressing galanin and recording from the galanin GABAergic neurons. Interestingly, since the galinergic neurons are also GABAergic, there is no communication between the galanin ones. So there is really no reciprocal inhibition. It is important because it was important to test where the source of GABA comes from which GABAergic group in the VLPO. And again, this is like a release of GABA, goes away with a GABA antagonist, and is maintained in the presence of monosynaptic condition. Now, this is now the end of the story to demonstrate that we have a switch in the VLPO. Again, I try to recap a little bit. So we have 2 groups of neurons. They communicate between each other. And the orexin is exciting one cell type that is not expressing galanin and inhibits the other cell type that is GABA and galanin, but indirectly through this local input. But how we can show this? We need to combine all together. We need to combine the optogenetics, the pharmacology, and then the single cell identification. And I did this in the last very complicated experiment in which I stimulated, again, the input locally in the VLPO from the GABA non-galanin to the GABA-galanin, and I applied orexin in the bath as a drug, of course, as always, but keeping activated my local input. What I saw, it's an increase of this evoked current. It means that the effect of orexin inhibiting these galanin neurons comes from the local GABAergic input, because once I have this switch on, I can modulate the activity of the switch with something that really potentiates the activity of the switch. And orexin goes and activates this local input that is activated and keeps more activated, and also keeps more and more GABA to be released. And it's calculated by this parabolic analysis. And we showed here that is not much change the unitary current, so the conductance is not really much involved, but the number of GABA channels, yes. So we have more GABA that has to go on more channels, and we have a lot of transmission that is like enhanced by the orexin application. So now we go to the molecular, advanced molecular, I called all the time, because now we simply showed a circuit. We simply showed two groups that basically they are different, but they're different only from the galanin expression. But I never believed that. I believe that these neurons, they share similarities, but they can be much more different. And the only possible way to show the difference was to go and use the RNA-seq data from the preoptic area, published by Moffitt and Dulac et Zhuang, and through the Murfish. So what we did with the help of my colleagues was to reconstruct the VLPO molecular organization. But of course, we started from the preoptic area, that is really the bigger area. VLPO is just a part of the preoptic area. So to build up the VLPO atlas, we had to retrieve and clusterize again the VLPO. And as you can see here, we used a pipeline that was capable of reproducing exactly the results from Moffitt et al. So it means that our bioinformatic approach was correct and was beautifully done. And the only possible way to reconstruct something small from something big was through the spatial transcriptomics. So we took all the clusters mapping into the VLPO, combined together, and building up the VLPO atlas, that is like A and B on the bottom right. So from the preoptic area, we have the VLPO after analyzing only the clusters mapping in there through the Murfish. Murfish is, as most of you know, is like a multiplexed error-robust in situ hybridization, and this relies on a combinatorial analysis and through a specific binary barcode that can label a specific transcript of specific genes. It's a very powerful approach that can give to you the information about the amount of the transcript that is like expressed. So you can make clustering, you can make like a quantification, and also the spatial resolution, the spatial location of this transcript. And it was so important for my study because I could really demonstrate my findings through the data from other people. But the new thing here is that I was able to reconstruct the VLPO from the preoptic area, because these people worked on the preoptic area. I was only interested in the VLPO. This is the ventrolateral part of the preoptic area. So we have now in front of us really two groups of neurons. Now I believe that there are really two different groups of neurons, because we took from the VLPO cluster only the GABAergic neurons, and we did a differential expression between the VLPO GABA versus the VLPO GABA galanin, and we found that they have similarities, but they show also many differences. And they are belonging to different populations. And of course, in panel B, you can see that the VLPO GABA is not expressing galanin compared to the other one, but they both express VGAT, so the capability of releasing GABA. They're producing GABA enzymes. They don't show the capability of releasing glutamate, so they're not excitatory. And they don't show the orexin receptor 1, but they show the orexin receptor 2. This is our last finding. And it was so important to see this because the orexin receptor 2 is the major target that people study in narcolepsy. And of course, you can see here from the graph that is expressed by the second bigger cluster in the VLPO GABA population, and is absent in the galanin neurons. And particularly is like only this cluster in the VLPO that express the high amount of orexin receptor 2, the cluster 0. And I verified this through my RNA scope in situ hybridization. So I did 3 genes. Here is a triple RNA scope in which I labeled the galanin, the VGAT, so the capability of being GABAergic, and the orexin receptor 2. As you can see here, the galanin are also GABAergic. Of course, we know this many times, so I showed this. And the GABAergic neurons, non-galaninergic, express the orexin receptor 2. Not all of them, but a nice fraction of them express the orexin receptor 2. It means that when orexin is in there, activates those neurons, and those neurons inhibit downstream monosynaptically the sleep-promoting galanin neurons. And this is the circuit that promotes arousal, stopping sleep. So in this last part of the talk, I'm going to show you the last experimental slide in which I can prove that functionally the orexin cells send functional projection to the VLPO, and this, interestingly, not only orexin can be released but also glutamate, because orexin neurons are glutamatergic. And I did this through, again, using orexin cream mouse. So I stimulated the fibers in the VLPO, I recorded the VLPO neurons, and I can see that orexin neurons can release, together with orexin, also glutamate and activate the AMPA receptors. And the question now is, where this transmission goes on the galanin? No, again, goes on not galanin neurons, because if we want to have arousal, we need to stop the galanin, not activate them. And as orexin, glutamate goes in the same direction. So the stimulation of the orexin VLPO pathway is capable of releasing glutamate on the neurons that they are excited by noradrenaline and they lack galanin expression. So, to conclude and to summarize a little bit our findings, we found that orexin neurons promote wakefulness by inhibiting the sleep-promoting neurons GABA-galanin in the VLPO. And this is a feed-forward inhibition, so it's an indirect mechanism. In vivo optogenetic stimulation of orexin input in the VLPO promotes rapid arousal from both stages of sleep, REM and non-REM. We found a dual response of orexin in the VLPO in vitro, so as a drug, orexin excites and inhibits. But remember, the inhibition can be only indirect, because through the receptors that we know, the orexin can only excite. If any inhibition is detected, is indirect and mediated by something else. So the excitation is direct and inhibition is mediated by GABA-A signaling and is indirect. And more importantly, the orexin inhibits the VLPO GABA-galanin neurons by feed-forward mechanism through the increase of the GABAergic afferent inputs. And these inputs is coming from, in part, from the local inhibitory input in the VLPO. So stimulating the local GABAergic neurons, VLPO GABA. And of course, also orexin neurons are capable of releasing orexin and glutamate. And we found through the RNA sequencing analysis and MERFISH that VLPO GABA and GABA-galanin neurons belong to different neural populations. Our future aim would be to now study the switch, study the VLPO GABA neurons, because we found out the markers to manipulate them separately and excluding from, contaminating with the galanin ones. And once we know a specific marker, we can really do specific manipulation and also treat them specifically with some drug or whatever can be also the next project. So the most important thing was to distinguish between these two groups, because they share many things, and so far it's impossible to discriminate them. But now we know the markers through the single cell RNA reanalysis and the MERFISH study. And this is my scheme. Oh, sorry. I apologize. Okay, this is my scheme for the conclusion. So we have two circuits discovered here. We have an orexin VLPO input and the VLPO GABA-galanin internal circuit. As you can see here, orexin send projection everywhere in the brain, also in the most important monoaminergic groups that trigger arousal. So orexin enforces arousal. Orexin is important in stabilizing wakefulness, and through this, it's involved the serotoninergic neurons of the dorsal raphe, the locus ceruleus neurons that release noradrenaline, and the T-main neurons that release histamine. But we found a new projection. I mean, was already known, but we found basically the mechanism, so the functionality and what was going on in there. And this projection of orexin into the VLPO is a novel and is acting on an internal population in the VLPO that is not galaninergic, but is GABAergic. So when we shine the light here, we can induce the release of orexin and glutamate that can activate the local GABAergic neurons through the AMPA receptors and most importantly, orexin receptor 2. And once these neurons activated by orexin and glutamate release GABA, they shut down the galanin neurons in the VLPO. So from the lateral hypothalamus, we have two groups so far that send projection to the VLPO, to published studies. One, if you remember, it's GABAergic from the LH, and it's shutting down monosynaptically the galanin. One is the orexin and shuts down the galanin through a presynaptic mechanism through the local GABAergic neurons in the VLPO. And VLPO is like becoming very complex now. So we know that there are two groups, at least two groups of GABAergic neurons. I mean, the groups can be more if you follow basically the differential expression and the expression of the genes. But to summarize, basically, functionally speaking, now we can, and to make also the model simple, we can assume that we have mainly two groups in there. One that is like wake or involved in wakefulness, the other one that is like involved in sleep. And the one that promotes wakefulness is the interneuron, shuts down the local galanin cells that promote sleep. And the next step of my work would be to identify the other pathways, other circuits that modulate this. Now we know the LH-GABA that is directly here. We know the orexin that is like acting here. And we also will look more about the other sleep pathways. So how the sleep pathway can disinhibit the galanin neurons and shut down the local GABAergic neurons that are promoting wakefulness. So how we can promote sleep in this case instead of arousal. And we propose here a new role of orexin. Orexin, we know that can increase the input of the monoamines in the VLPO, releasing more histamine, releasing more noradrenaline, releasing more serotonin. And we know now that orexin has also a local effect into the VLPO through an internal input. And what I'm doing now next with my next study is to study this in the narcolepsy through a narcolepsy-like model in which we abolish the orexin neurons and see how the transmitters, the monoamines in this case in the VLPO, can change. This is a very, very, very important. The next project will be more important than this one because it can give us an idea how to basically face the disease in terms of, okay, what's going on in terms of monoamines, what's going on in terms of transmission release, and what are the targets that can really study with the specific drugs. So I would like to thank, of course, the organizers of the webinar, GenomeWeb and VizChen, to give me this possibility to present my recent work that hopefully will be out soon in Nature Communication because it's accepted. And I would like to thank also people in the lab that helped out a lot in this project in these years. My mentor Elda Rigoni and Patrick Fuller, and Dr. Grace that did the in vivo stimulation together with Dr. Benner, and people from Skemu Lab and Saper Lab and Ramalingam that gave us specific mice to do these specific manipulations. And Stefano Nardone from Lowell Lab that did also the analysis and interpretation of the data of single-cell RNA sequencing and MERFISH. Antonino also gave bioinformatic support, and other people that also provided the orexin-3 mice. And you for your attention. Thank you very much.
Thank you, Roberto. As a reminder, webinar participants, if you have a question, please type it into the Q&A box in the control panel. We'd also like to ask attendees to take a moment after the webinar has ended to take our exit survey to give us your feedback. And so we'll now start the Q&A portion of the webinar. And the first question is, how many genes did you target with your MERFISH measurement?
Okay, so yes, I would like to come back to to the specific slide. Okay, 1 second. So basically, when we started this work, it was very difficult to imagine the VLPO without having in front of us the preoptic area, that is the biggest, the bigger region, so where we can isolate the VLPO. Actually, when we clusterize the VLPO through MERFISH, we can have in front of us 20 clusters. And in these clusters, so we have different populations. Also, we have the glutamatergic population. I didn't analyze the glutamatergy because I was not interested in having other groups of neurons that will complicate more my model. I was more interested in having in front of me the GABAergic inhibitory neurons. And when we have the atlas of the VLPO through MERFISH, we move to the differential expression between the GABAergic neurons. And I plotted here only the top 15 hyper and hypo differentially expressed genes. Just an example. Well, we have many more. I cannot remember now how many, but for convenience, the figure we just plotted the 15 that are hyper and hypo expressed. And these two groups are really different. I mean, they share many things, but we can now have the specific markers to go through and to isolate them once for all, and we can also induce our manipulations better.
Let's see. What other pathways do you think might be involved in promoting wakefulness and arousal? What are you looking at there next?
Yes, it's a very important question. So I would like to go back to my last scheme that helps a lot. So basically, for the moment, we published from the lateral hypothalamus two big inputs in there. And if you remember why we have chosen the lateral hypothalamus, because from the tracing study, we found that this area sends a lot of inputs in the VLPO. Of course, in the lab, we also explored other things, like also the TMN histamine neurons input in the VLPO. But basically now we have an idea that the LH that is really involved in wakefulness can shut down the VLPO at least in two ways. One, it's monosynaptically through the GABA release, and the other one through orexin activating a GABA internal circuit in the VLPO. Now I'm studying the noradrenergic input in the VLPO. So I'm writing the manuscript. I need to finish the last experiment, but in this study, I will use also a narcolepsy model. So as I mentioned at the beginning, we are slowly killing the orexin neurons, and we measure the basically how the tone of noradrenaline can change in the VLPO compared to a wild type situation. So before the lesion of orexin neurons, where the mouse is okay, there is like a noradrenaline tone during wakefulness. But what happens if I basically kill the input to induce the noradrenaline release, and the input drops down? It's a very, 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 very important discovery, because it's like we have now a more meaningful idea about this region in the stabilization of the sleep and wake states.
And do you think that your findings at this point or in the future will point to potential treatment or treatment targets for narcolepsy or other arousal disorders?
I think so, because now when we have in front of us basically the differential expression, so we have a panel of genes that we can really look through, and of course it's important to mention that the RNA world is different from the proteomic world. And indeed, in this study, I started with the pharmacology first, and moving, of course, from the proteomic functional world to the RNA world. The two sides, they compensate each other. I mean, it's important to have at least in front of us an idea about the expression of the genes, but also it's important to have as a final target what messenger RNA can have a functionality. So this messenger can be converted into a protein. This messenger mRNA can be converted into a channel or like a receptor that can manipulate with a drug. So actually, we need both. We need the starting point, in my opinion, if you want to do like an unsupervised study, it's to have like RNA expression, so the gene expression. Then at the end, you want to narrow down to a true expressed mRNA family to have like in front of you a real target to manipulate with a drug. Yeah. And in this case, I think finding the GABAergic switch in the VLPO was so important because now we have like a common entry, or like, you know, a common switch for many points of entry in which many things can work, and the situation can really change. So yes, I'm expecting that we can really extend our study in this direction with a functional targeted study with the pharmacology more in detail, only focusing on the one group of neurons that is really different from the other one.
Well, that looks like all the questions we have for today. So we'd like to thank Roberto De Luca and our sponsor, VizGen. As a reminder, please look out for the survey after you log out to provide your feedback. If you missed any part of this webinar or would like to listen to it again, an archive version will be emailed to all attendees. Thank you for joining us for this genome webinar.
Thank you.